Our experience of the outside world is encoded in patterns of neural activity. Photons impinging on our retinas or pressure waves reaching our ears are each converted into patterns of neuronal activity that allow us to recognize the face of a friend or the sound of a barking dog. Similarly, the dynamics of neuronal activity in our brains enable us to reach for a coffee cup or type on a laptop. The basic goal of neurobiology is to understand how all of this happens. How does the brain encode sensory information, store memories, and allow us to move around and interact with the world? Moreover, the goal of developmental neurobiology is to understand how complex nervous systems capable of carrying out these tasks are able to self-assemble during development. Fundamental to each of these objectives is the ability to actually visualize the activity of large numbers of neurons in the brain as an animal processes sensory information or engages in motor behavior. At present, one of the most powerful and most widely used approaches to visualizing neural activity on a large scale is calcium imaging. At rest, intracellular levels of calcium are very low. When a neuron is active, the change in membrane potential opens voltage-gated calcium channels, allowing calcium to flow into the cell. A variety of cellular pumps and buffers clear this elevated calcium. Thus, the transient increase in intracellular calcium levels is temporally coupled to neuronal activity and provides a reliable, indirect indicator or proxy for this neuronal activity. To exploit this calcium signal, we introduce a gene into neurons that encodes a protein that has been engineered to exhibit enhanced fluorescence upon binding calcium ions. In the absence of calcium, the absorption of a blue photon by this calcium indicator results in little or no fluorescence. However, when the calcium indicator binds calcium ions, shown here in purple, the absorption of a blue photon results in robust fluorescence. Increases in fluorescence are indicative of neuronal activity. While calcium imaging is widely employed in model organisms as diverse as C. elegans and primates, our lab uses it to study neural network assembly in developing zebrafish larvae. The advantages of zebrafish are that the larvae are small, transparent, and develop rapidly. It is possible to image the entire brain, or nearly the entire brain, while providing well-defined sensory stimuli to the larva. We start by embedding the zebrafish larvae in small drops of low melting point agarose. The embryos are briefly immersed in the melted agarose. Then a small drop with the larvae is placed in a custom-built imaging chamber. Then the larva is oriented using a fine gauge fishing line before the agarose is able to harden. Once the agarose is fully solidified, the imaging chamber is then filled with medium to immerse the immobilized zebrafish larva. Zebrafish embryos and larvae can be maintained in agarose in this way for several hours. They can be removed from the agarose after imaging and allowed to develop further. This enables the same fish to be imaged on multiple days. Once prepared, the imaging chamber with the immobilized larva can be placed on the stage of an upright, custom-built 2-photon microscope, which we use in conjunction with water immersion objective lenses. To understand the development of the neural circuits that process visual information, as well as the genetic basis of assembling these circuits, we can image neural activity while providing well-defined visual stimuli. We provide these visual stimuli using a picoprojector, shown here to the right of the microscope. Images are projected onto a diffusive screen positioned a few centimeters from one eye. To prevent contamination by stray light, the projector is enclosed in a light-tight box, as shown here. To prevent light contamination from the computer screen and other light sources in the room, the entire microscope is housed in a light-tight enclosure. In this example, we show two optical sections from a typical movie. The field of view shows the midbrain and hindbrain with the head oriented to the left. The small flashing dots represent the activity of individual neurons. From such movies, we can measure the activity in a large number of neurons throughout the brain, as shown here. In this experiment, we identified over 2,500 active neurons, with each fluorescence time course represented as a row in this matrix. The bright yellow color represents elevated fluorescence relative to the darker blue color. Calcium imaging has significant advantages for monitoring neuronal activity in vivo. First, large numbers of neurons can be imaged simultaneously, providing a more complete view of neural dynamics. Second, the approach is very flexible, as calcium indicators can be targeted to specific cell populations or neuronal types, and they can be fused to other genes to localize them to different subcellular compartments. In addition, calcium imaging is relatively easy to implement, as anyone with access to a 2-photon, confocal, or light sheet microscope could get started in principle, although there is a significant learning curve associated with data analysis. However, calcium imaging also has some disadvantages. Most notably, the temporal resolution is much slower than electrophysiology, with the response of the neuron smeared out in time compared to the sharp, discrete spikes obtained in electrical recordings. In addition, using modern genetically encoded calcium indicators requires introducing them into the animal, either through transgenic approaches or through viral expression.